All right, let's get back to talking about insects and exoskeleton, or the integument, or the cuticle. Like I said last time, everything that you see on the insect here, everything that's visible, is constructed out of the cuticle. So, and that includes the eyes, the entire mouth, all of the exoskeleton, and the first and last parts of the digestive system, which are called the foregut and the hindgut, as well as all of the breathing tubes on the insect, which are called trachea. So these are constructed of this hard material, the sclerotized chitin. Chitin itself is not a hard material, but once it becomes sclerotized, it becomes very rigid. And so as the insect grows, at some point it has to shed this exoskeleton so that it can progress to the next stage of growth. Otherwise, it will kind of be smothered within its own skin. So how the insect accomplishes this is through a process of ecdysis, which is the shedding of the old exoskeleton. How does an insect know when it needs to shed its exoskeleton? So the soft parts of the insect continuously grow and press up against the exoskeleton, and at some point this triggers a series of nerves called the stretch receptors that the insect has become too big for its own skin or its own cuticle. At this point, it will begin the cuticular shedding process. So this is the same diagram that I talked about last time. This is what the cuticle looks like in cross section. So at the bottom here, you have the basement membrane, which supports everything, and the epidermis, the living tissue, the living cells are on top of this membrane. There's a small gap where usually the formation of new chitin is being made. Above that, you have this soft chitin layer called the endocuticle. Above that, you have the exocuticle, which is the hardened, sclerotized, melanated part of the cuticle. This is the quote-unquote shell of the insect. And then above that, you have the epicuticle, which is a wax layer which protects the insect from dehydration and some parasites and things like that. So when the insect becomes too large, when it realizes it's too large for its own cuticle, the first thing it does is apolysis, or apolysis, if you want to call it like that. This is the separation. Apolysis is the separation of the old cuticle from the epidermis and the basement membrane, and the formation gap there gets wider. Once that formation gap gets wider, the first thing that happens is the insect lays down a new epicuticle. And this is a wax membrane. And it lays down a new wax membrane between the epidermis and the old cuticle once apolysis has started. And this is because into this gap, digestive enzymes are going to be pumped in. So chitin is a carbohydrate, and it can be digested by enzymes called chitinases. All enzymes generally end in ACE, and it usually tells you what it is acting on. So chitinase is something that acts on chitin. In this case, it digests it. So it lays down a new wax layer, this new epicuticle, and it pumps digestive fluid into this gap above the wax layer. That digestive fluid will then become active, and it will begin eating away at this old endocuticle. It'll begin eating away at this and reabsorbing it. The insect will then reabsorb all of this chitin and all of these proteins that are in here to be reused. It completely destroys the endocuticle. And while it is destroying that endocuticle, it is slowly building up new cuticle in this area here. And it builds from the bottom, and it will in this case from the top down, but it will continuously secrete new chitin and new cuticle into this area here. Once all of this digestion is done, it begins the ecdysis process, which is the actual shedding of the old cuticle. And the insects do this by creating pressure, sometimes ballooning up, sucking in air. They sometimes have special organs on the body or structures on the body that help them shatter the old cuticle. And they shatter the old cuticle along points on the insect called ecdysial lines. Again, the word ecdysis. And these are weak points in the exocuticle. So in this case, at random, not at random points, but at very specific points on the body, the insect will have kind of a thinning of the cuticle. There'll be a thinning point here and a thinning point there, so that when the insect actually begins to shed, these points are very weak, and these are ecdysial lines, and they'll shatter as the insect expands. And then it just crawls out of its old exoskeleton. In this case, it can be kind of traumatic. When an insect emerges from the exoskeleton, it hasn't completely formed the new cuticle. It's still very soft, so this cuticle down here is still being formed. And that's why when you see like a butterfly come out of a cocoon or a cicada come out of its nymphal shell, it's very pale. It hasn't developed the melanin colorization yet. It's very soft. It hasn't hardened itself with the sclerotized exocuticle yet. It's very fragile in this state. So it has to sit there. With butterflies, sometimes they're drying their wings. They're not drying them, they're hardening them and expanding them. So during this stage, it's very precarious for them. They're very sensitive to pathogens. They're very susceptible to predation. After a couple hours or even a couple minutes for smaller insects, it will completely sclerotize a new cuticle layer and it can go about its day. So it'll re-sclerotize things. When it comes out of its old cuticle, it also has to pull out the old cuticle lining of its foregut and hindgut, as well as the cuticular lining of all of its trachea. So it's basically pulling out of its mouth and out of its butt all of the old digestive system that it's trying to shed, and kind of ripping out its own lungs in the form of the trachea, and then regrowing those tissues and resetting them with new cuticle. So this is an extremely important process for insects. The other reason why this ecdysis process is important is because we use the shedding of the old exoskeleton to mark developmental stages. And the most common way that we mark these is through a system called the instars. If I can spell, which I can't. Instars. And these are developmental stages of an insect. So an insect generally starts out as an egg, which is laid, as opposed to sometimes insects will give live birth. And let's say that this is a fly. So it ends up as a small maggot, and it sheds its skin and it becomes a larger maggot, and it sheds its skin and becomes an even bigger maggot. And then it becomes a big fly. This is my terrible fly drawing, right? And its big eye. So, each of these we call an instar. So this is the first instar. This is the second instar. This is the third. This is the adult. So each of these represents a molt, or the ecdysis of the old exoskeleton. Generally, with each species of insect, the number of molts is a fixed number. You won't have a random number of molts. So like with crustaceans, like lobsters, they can continue to grow and they'll continue to molt over the course of their entire life. Insects don't do that. They have a fixed number, generally 4 to 5. A lot of beetles and flies and things only have 3. But that's how we measure development in insects. So this would be the egg. So if you had a first instar larva, everyone would know, if you said a first instar, that this is the larva that just hatched from the egg. And it's a universal code. The other way that things are measured is through stadia, which is kind of an old term. And instead of instead of each individual life stage like this with an instar between molts, it's actually across molts, and we call that the stadium. It's not used as widely as instar. Go with instar, and you everyone will understand you. So that's all I'm going to talk about for ecdysis today. Next time, I think I'm going to move on to the insect head and the structures of the head and how the mouth works, and that is the point in morphology where things start to become very complicated. So this is the easy stuff. Now we're going to move on to the hard stuff. All right, thanks. Until next time.